Hi everybody, and welcome to our module on the TCA cycle. The TCA cycle is a very important metabolic pathway in human cells. TCA stands for tricarboxylic acid cycle. It's also sometimes called the Krebs cycle, named after the person who discovered it, or the citric acid cycle. And this cycle will convert molecules of acetyl-CoA into molecules of CO2, and in doing so it will derive energy from the reactions. All the reactions of the TCA cycle occur in the mitochondria, and this is very convenient because some of the molecules that the TCA cycle produce, like NADH and FADH2, are used by the electron transport chain to generate ATP, and the electron transport chain is also found in the mitochondria, so these molecules are therefore synthesized right where they need to be used. In addition to NADH and FADH2, the TCA cycle produces molecules of GTP, and then, as I said before, it produces carbon dioxide. This slide shows the intermediates of the TCA cycle, and we'll go through each of these reactions one at a time in the remainder of this module, but I want to make an important big picture point before we get started. The TCA cycle begins with a molecule of acetyl-CoA donating 2 carbons to the TCA cycle. 2 carbons are then removed in the form of CO2 at this step here and this step here. This means there is no net increase in the amount of carbons in this cycle when a molecule of acetyl-CoA enters. And this has an important implication for the generation of glucose via gluconeogenesis. You may know that oxaloacetate can be shunted to glucose via gluconeogenesis. Well, in order to do this, you need to increase the amount of oxaloacetate present in the TCA cycle, and acetyl-CoA will not do that. It will donate 2 carbons will leave, and so however many molecules of oxaloacetate you had when the acetyl-CoA entered will be the same number you have when it leaves. You won't have increased the amount of oxaloacetate, and therefore the acetyl-CoA cannot be converted into glucose. This has important implications for when we talk about things like fatty acids. Even chain fatty acids can be converted into acetyl-CoA. This means they can be used to derive energy, but they cannot be used to derive glucose. The fatty acid will be converted to acetyl-CoA. The acetyl-CoA will donate its carbons, and the carbons will leave in CO2. There'll be no increase in the amount of oxaloacetate, and therefore there'll be no ability to use the acetyl-CoA from the fatty acids for gluconeogenesis. That's an important concept, and make sure that makes sense to you as we go forward. So now we'll go through the reactions of the TCA cycle one at a time, and the first one involves synthesis of a molecule of citrate. This is the structure of citrate at the bottom of the screen here. It is a 6-carbon structure, and it is synthesized from a 4-carbon molecule of oxaloacetate, which I've shown on the screen here, plus 2 carbons that come from acetyl-CoA. The enzyme that catalyzes this step is called citrate synthase, and that should make sense to you because it synthesizes citrate. This is an irreversible step, and as we shall see, the irreversible steps are where all the regulation occurs. In this case, the regulation occurs via ATP. When ATP levels get high, that will inhibit citrate synthase and slow down this step of the TCA cycle. You should know that citrate is a very important indicator of the energy level inside of a cell. The subsequent steps in the TCA cycle are inhibited by high levels of ATP. That will slow down the metabolism of citrate and cause the level of citrate to climb when levels of ATP inside of a cell are high. Therefore, the cell will use citrate as a marker of high energy levels, and when citrate levels are high, it will inhibit other pathways. Citrate can inhibit glycolysis by inhibiting phosphofructokinase 1, and this should make sense to you. When ATP levels are high, citrate gets high, and therefore the cell does not need to do glycolysis. In addition, when citrate levels get high, this will activate acetyl-CoA carboxylase, which is an important enzyme for fatty acid synthesis. When citrate levels get high, it signals that there's lots of energy around, and therefore the cell wants to begin storing that energy in the form of fatty acids. There's an important clinical correlation of this step of the TCA cycle, and that involves fasting. When patients are in the fasting state, oxaloacetate is used for gluconeogenesis. I've shown you here how pyruvate can be diverted to oxaloacetate and then into glucose via gluconeogenesis. This lowers the level of oxaloacetate available for citrate synthase to use, and as a result, less acetyl-CoA is pulled into the TCA cycle. In addition, in the fasting state, lots of acetyl-CoA is being synthesized from fatty acid beta oxidation. So for both of these reasons, you get very high levels of acetyl-CoA inside of the liver in the fasting state. When this happens, the liver will then synthesize ketone bodies, which can be used as an alternative energy source. And this is the mechanism by which ketone body synthesis is triggered in the fasting state. Isocitrate is an isomer of citrate. That's how it gets its name. It has the same chemical structure. The molecules are just arranged differently around the carbon skeleton. You can see that this OH group is attached to the central carbon in citrate. It's moved down one carbon in isocitrate closer to the end. The enzyme that catalyzes this step is called aconitase. It actually forms an intermediate called cis-aconitate, and then it forms isocitrate. This enzyme can be inhibited by a substance called fluoroacetate, which is used in rat poison. And this is a reversible step, and as I said before, it's the irreversible steps where all the regulation occurs, so there isn't any important regulation of this step. Next, in the TCA cycle, we form a molecule of alpha-ketoglutarate. Alpha-ketoglutarate is a 5-carbon structure, as I've shown here, and since we derive it from a 6-carbon structure of isocitrate, we have to remove a carbon, and we do that in the form of CO2. This step also produces our first molecule of NADH, which can go to the electron transport chain to generate ATP. The enzyme that catalyzes this step is called isocitrate dehydrogenase, and this is one of the rate-limiting steps of the TCA cycle. This is an irreversible step, and as I said before, the irreversible steps are where all the regulation occurs. Isocitrate dehydrogenase is inhibited by ATP and NADH, and these molecules will be present in greater amounts when there's lots of energy inside the cell, and therefore it wants to inhibit this step and slow down the TCA cycle. Isocitrate dehydrogenase is activated by ADP, so when ADP levels are high, that means lots of ATP has been used, and the cell needs more energy, so therefore it activates this step of the TCA cycle. And in addition, this step can be activated by calcium, and that's very important. In skeletal muscles, they want the TCA cycle to start going as soon as they are contracting, and the calcium that gets released in contracting muscles will therefore trigger this step so that the TCA cycle begins to work. The next step involves synthesis of a molecule of succinyl-CoA, and this is done by a complex called the alpha-ketoglutarate dehydrogenase complex. This is a complex of 3 enzymes just like the pyruvate dehydrogenase complex. In a separate module, I discussed the PDH complex and how it works with 3 enzymes functioning together in concert with 5 cofactors: thiamine, CoA, NAD, FADH, and lipoic acid. Alpha-ketoglutarate dehydrogenase complex works the same way and requires the same cofactors. It takes a molecule of CoA and adds it to alpha-ketoglutarate to create succinyl-CoA. In doing so, it removes a molecule of CO2, and therefore we go from a 5-carbon structure to a 4-carbon structure. This step also produces our second molecule of NADH from the TCA cycle, which can go to the electron transport chain to generate ATP. This is an irreversible step, and therefore regulation will occur here just as it does in the other irreversible steps. This step can be inhibited by succinyl-CoA. This is feedback inhibition, so when there's lots of succinyl-CoA around, it will slow down this step. It's also inhibited by NADH, so when there's lots of NADH around, this step will be slowed. And then also, just like the last step in the TCA cycle, calcium can activate alpha-ketoglutarate dehydrogenase to make the TCA cycle move faster when skeletal muscle is contracting. The next step involves the formation of succinate. This is done by an enzyme called succinyl-CoA synthase. It removes the CoA portion of succinyl-CoA, and in doing so, it generates a molecule of GTP, which can be used just like ATP for energy. The next step involves formation of a molecule of fumarate. The enzyme that does this is called succinate dehydrogenase, and this is a unique enzyme because it's embedded in the mitochondrial membrane, and it also functions as complex 2 in electron transport. So it's an enzyme for both the TCA cycle and the electron transport chain. It converts succinate into fumarate. This is a reversible reaction, and in doing so, it generates a molecule of FADH2, which can immediately go into the electron transport chain. So complex 2 and succinate dehydrogenase are the same enzyme, and they function in both pathways. Fumarate is a tie-in between the TCA cycle and several other pathways. The urea cycle can form fumarate, which can then be fed into the TCA cycle. In purine synthesis, fumarate is a step in formation of IMP, and you can also generate fumarate from the breakdown of amino acids, especially phenylalanine and tyrosine. So all these different pathways feed into the TCA cycle by feeding into fumarate. The next 2 steps are reversible. Fumarate is converted into malate by an enzyme called fumarase, and then malate dehydrogenase converts malate into oxaloacetate, and in doing so, it generates another molecule of NADH, which can head to the electron transport chain and be used to generate ATP. This reversible step of converting malate to oxaloacetate can be used to shuttle molecules from the cytosol into the mitochondria. This is called the malate shuttle. In order to understand the malate shuttle, you need to understand two key points. The first one is that malate can cross the mitochondrial membranes via a transporter. NADH and oxaloacetate, however, cannot cross. So the malate shuttle is going to be used to transport NADH into the mitochondria for use in electron transport, and also transfer oxaloacetate out of the mitochondria for gluconeogenesis. Let's talk about this in a little bit more detail. So use number one of the malate shuttle is for the transfer of NADH into the mitochondria. So let's imagine we have a molecule of NADH and it's in the cytosol. It needs to get into the mitochondria to participate in electron transport, but it cannot cross the membrane. The way we're going to get it across is by using the malate shuttle. So we will synthesize a molecule of malate from oxaloacetate. In doing so, the NADH will be converted to NAD+. Essentially what we've done is donate the electrons from NADH to malate. Malate can then shuttle across the mitochondria, bringing those electrons inside the mitochondria, and then the malate can be converted back into oxaloacetate. In doing so, it can give its electrons to NAD and create a molecule of NADH. So you can see that as a result of this shuttle, the NADH, which was stuck in the cytosol, has now been moved inside into the mitochondria, where it can participate in electron transport. Now we want to regenerate the oxaloacetate that we used on the outside of the mitochondria. To do that, we'll convert the oxaloacetate inside the mitochondria into the amino acid aspartate. This involves conversion of the amino acid glutamate into alpha-ketoglutarate. The aspartate can then go back across because it's able to traverse the mitochondrial membrane, and on the other side the reaction can be reversed, and thus we have regenerated the oxaloacetate that we used in the beginning of this process. The second use of the malate shuttle is the transfer of oxaloacetate out of the mitochondria for participation in gluconeogenesis. So to understand this, let's imagine that we have a molecule of oxaloacetate. It's inside the mitochondria and it wants to get out so it can participate in gluconeogenesis. How is it going to do that? Well, first we'll convert it to a molecule of malate using that reversible reaction of the TCA cycle. The malate can then cross into the cytosol, and then the reaction can be reversed in the cytosol, and now we have oxaloacetate outside of the mitochondria for participation in gluconeogenesis. So now we've gone through all the reactions of the TCA cycle. On this slide, I just want to point out to you that many of the TCA intermediates are used in other metabolic pathways. Citrate is very important for fatty acid synthesis. Alpha-ketoglutarate is involved in the metabolism of many amino acids. So is oxaloacetate. And then, as I showed you on the last two slides, oxaloacetate can be used to synthesize glucose via gluconeogenesis. Succinyl-CoA is also a very important crossroads of many different metabolic pathways. You can get succinyl-CoA from alpha-ketoglutarate in the TCA cycle, as we just discussed. It can be converted into succinate via the TCA cycle. In addition, you can get succinyl-CoA from methylmalonyl-CoA, and methylmalonyl-CoA can be derived from odd-chain fatty acids and branched-chain amino acids. And then finally, succinyl-CoA is a very important substrate for the synthesis of heme molecules and hemoglobin. I'll finish with a few summary slides. Here are all the inhibitors of the TCA cycle: ATP, NADH, acetyl-CoA, citrate, and succinyl-CoA. All of these things rise inside of cells when there are lots of energy sources available, and therefore the cell wants to slow down the TCA cycle. On this slide, I've shown you where all these inhibitors exert their effects. So ATP, acetyl-CoA, and NADH can inhibit pyruvate dehydrogenase so that pyruvate is not converted into acetyl-CoA. ATP and citrate can inhibit citrate synthase. ATP and NADH can inhibit isocitrate dehydrogenase. And then finally, NADH and succinyl-CoA can inhibit alpha-ketoglutarate dehydrogenase. The TCA cycle is activated by ADP and calcium. Here's where those activators exert their effect. ADP and calcium can both activate isocitrate dehydrogenase, and calcium can activate alpha-ketoglutarate dehydrogenase. And that concludes our module on the TCA cycle.